they have this one set of genetic material that they copy and they pass on. So how does this sort of immense variety of, genetically distinct strains within a single species or multiple different species we see? There must be some way that these microbes are evolving genomically. So we're going to look at how these mutations arise within these species that help set apart maybe certain subspecies from each other. And of course, you can have a cumulative effect of multiple mutations, which will, set organisms apart. So generally within bacteria, it's sometimes very difficult to determine what's a separate species versus a strain of an existing species. So Salmonella for the longest time, is considered its own species, but then, you read some other literature and it seems as though Salmonella is a subspecies of, E. coli. And so it's sort of not very easy to determine. Generally, anywhere from, about average of 85% to 90% of the DNA similarity means that this is the same species in bacteria. And, of course, it's a strain within that species. So we're going to talk about some mutations that would lead up to accumulation of enough mutations so that the organisms can begin to diverge into their own individual, species. So we're talking about mutations that are stable and they will be passed on to the daughter cells. And these can come in a variety. You can have point mutations where a single base pair might be changed. And then, of course, we've already studied how codons code for, amino acids. And if you have a single base pair change, it can potentially change the meaning of that codon into a completely different amino acid. And if that happens to be a critical amino acid to the function of the protein or the structure of the protein, it abolishes the function, it abolishes its structure. And so, cells may, for example, have a mutation in a critical gene that helps with the flagellum assembly, and you can have one cell that had flagellum, give rise to a daughter cell that lacks flagellum because of that single mutation. So they can be, quite critical. Similarly, you can have addition and deletion of base pairs as well, as we're going to talk about it. And then you can have, larger mutations, which are not so common in bacteria, but we're going to see that there can be certain elements that can go into bacteria circular genome, making a sort of a new novel mix of DNA, and that will of course, attribute to these larger mutations. Of course, anytime you have a piece of DNA go into an existing piece of DNA, and if that existing piece was a critical gene, then you can have mutation within that gene. If that piece was not so critical to the coding region or to the regulatory sequences, then it can be tolerated very well, and it doesn't have an effect on the phenotype of the organism. So, there will be some larger mutations that we'll talk about. And then majority of these mutations, of course, arise spontaneously as a byproduct of cells dividing so rapidly, right? E. coli, for example, divides every 20 minutes under ideal conditions. And so you have, replication events going on all the time. Things are growing at an exponential rate. And so you're going to have the rate of spontaneous mutation that is relatively low, but they do occur. And then there are induced mutations. These would occur in a laboratory setting where maybe a geneticist is trying to generate mutant bacterial strains to study newer phenotypes and connect them to genes that might have been mutated. So this is a way of really deciphering the function of individual genes. So we're going to look at, some methods of inducing mutations and how to detect mutants. So spontaneous mutations can occur during the process of replication. So, what are some of the things that can go wrong during replication? So I don't know, I never ask my students to memorize the structure of the nitrogenous bases, but it would be helpful to kind of go over the structure of the nitrogenous bases. Here you have a normal looking cytosine, and here you have what we refer to as the amino form of the cytosine. So the difference here is the bonding pattern of nitrogen to the ring structure. And as a result of that, you can have, varying number of hydrogen bonds that the cytosine is able to produce. So both adenine and cytosine occur in equilibrium with their, natural forms, as well as what we refer to as these amino forms. And in doing so, if either one of it is in its amino form, it is able to miss base pair. So if cytosine is in its amino form, it's going to base pair with the regular adenine, which is miss base pairing, because you expect it to base pair with, guanine, or you expect adenine to base pair with cytosine. And you will always have a double ring purine against a single ring pyrimidine, because that helps you ensure the diameter of the double-stranded DNA helix. So you will have that, but just the pairing partner has changed here. So generally it's adenine against thymine and cytosine against guanine, but here you are seeing that adenine and cytosine are base pairing. Okay, so that's misbase pairing. And then you can have the enol forms of either thymine or guanine, okay, that can miss base pair as well. Generally, thymine base pairs for base pairs with adenine, and it produces 2 hydrogen bonds. Here you're seeing that 3 hydrogen bonds are present between guanine and thymine. So regardless of which one of the two is in its enol form, they're going to miss base pair with each other. So if this was just, one cell and maybe there was miss base pairing, it might not be such a huge problem. The problem arises when you actually move on to the next generation, when this DNA strand that carries this base pair is copied. That is when the problem arises. So one of the things that RNA polymerase, DNA polymerase rather, does is it proofreads. So you ask yourself, like, why isn't it proofreading and saying that this is a missed base pair and I need to correct it? Because we know that DNA polymerase 3 has that, 3' to 5' activity where it backs up and it's removes the incorrect base and inserts a correct one and moves on. So why is this tolerated? So it turns out that, the DNA polymerases are essentially just looking for the number of hydrogen bonds, and they kind of related to one of the bases. So if they're just testing guanine, and right now guanine is base paired, and this has to be template, of course, and if it's base paired with a base that has 3 hydrogen bonds with guanine, then it's the correct base. Okay, so it doesn't distinguish between the bases on their chemical basis. Rather, it looks for the number of hydrogen bonds. If guanine was in the template and you have 3 hydrogen bonds that have been formed with the corresponding base, then it doesn't care whether it's the enol form of thymine or regular cytosine. So it considers it correct and it moves on, okay? But when time comes to copy that, okay, where maybe a error was made during replication and this erroneous DNA was passed on to the daughter cell, and now you have one strand where the sequence is essentially the same as the parental DNA, and whichever daughter cell inherits that, that's going to continue to be that original phenotype. However, the second daughter cell has received this mismatched base pair, and you can already see why this could be a problem. If the bottom strand was the template strand, it would, and it was for a critical gene, then this could already contribute to a improper amino acid in the protein sequence. But let's say cell is able to survive, and now during the second set of DNA replications, the bottom daughter cell is going to have normal replication, whereas the top daughter cell is going to use both of its DNA strands as template. The top strand would give rise to the same phenotype as the parental DNA. We'll call that the wild type phenotype. Whereas the bottom strand, when it serves as a template for replication of DNA, it's going to drive the insertion of an adenine against thymine. Okay, so right now we have a mutant strain here. And now this can go on and retain that mutation and pass it on to its own daughter cells as well. So, this resulted in sort of a transition mutation, right? So I believe that when we talk, so we have transitions where you go from, a purine to purine or pyrimidine to pyrimidine, and transversion is where you change it from one to the other. So we started with a pyrimidine, right? And it was changed into another, pyrimidine, so it's a transition mutation. Now, that's not the only way spontaneous mutations arise. Just having these various isomeric forms within of bases within the cells that we refer to as tautomers. You can also have spontaneous mutations arise at sites where you have multiple repeated sequences, right? So this here, repeated sequences are represented here as, for example, these sequence of adenines. Okay, so sometimes when DNA polymerase begins to copy these repeated sequences, it can make an error. So instead of inserting 1, 2, 3, 4, and 5, it can insert 6 or it can insert just 4. Okay, so why 4 versus 6 depends on where the error was made. If our DNA polymerase somehow miscalculated the number of T's that have already been inserted into the newly synthesized daughter strand, it will result in insertion of a new T, and that's represented by this green T. So this generally occurs when there is some sort of a slippage as the DNA polymerase is passing through. And, in this case, you're seeing just a single T that's been inserted, but you can have multiple repeats that may be looped out. So you can have multiple bases that might be, inserted. This is something that happens in human genetic disorders as well. So Huntington's disease results in expansion of repeats because of similar mechanisms. Okay, so it can happen within a bacteria as well. So if slippage occurs within the daughter strand, this bubbling out occurs within the daughter strand, then a additional repeat would be added. And so this will result in insertions. And of course, this would have to be stably inherited, passed on to the daughter cells on a subsequent round of replication. And this shouldn't be corrected during the replication processes. You can also have the same phenomenon occur where the slippage is actually in the template strand and rather not, within the daughter strand. And so the result is that you can have fewer than expected number of repeats that are added. So this is, again, happens in all sorts of organisms, happens within human cells as well. Duchenne muscular dystrophy has deletional mutations that can potentially be attributed to something similar. Okay, just to give you an example. So if the looping out or the slippage of the polymerase machinery happens to be on the template strand, then it's going to result in mutations that are equivalent to deletion mutation. And so insertional mutations and deletional mutations can be quite traumatic to the cell, as we're going to see in a moment. A third way spontaneous mutations arise is that sometimes within cells, there is depurination of nucleotides that occurs, which means that the backbone, the phosphodiester backbone of the DNA molecule is intact, but the base has been removed, and so there's no base pairing at that particular site. So here in this case, you're seeing from this particular sequence, the first G has been removed, which is a purine. So we refer to it as the apurinic site. You can also have a diminic sites as well, where pyrimidine has been removed as well. But so when this apurenic site undergoes replication, there is nothing to base pair with here. So DNA polymerase recognizes that there is, an opening there, and there must be something that goes in that spot, but it doesn't have a template to insert the accurate base against. So it has two choices. Either it just kind of waits there forever, hoping that something will change and then it can complete the rest of the replication, or it just randomly is going to insert one base and then continue to complete the replication and kind of rely on the proofreading mechanisms to come back and correct that. And that's what happens because there are multiple proofreading mechanisms within bacterial cells. So you're going to have, say, during the first round of replication, one of the template strands that gives rise to the same original wild type sequence, whereas the top strand is going to not have template at that particular site. So you can have any base really go in that site, right? So you can have a heterogeneous, mix beyond this particular replication cycle if it continues to happen, right? So you can have maybe first just, an A is inserted. And let's say if that apurenic site is never corrected and it stays, then every subsequent step of replication can randomly insert a brand new base there. So you can have, one time where you have an A there, you can have another time where it can serve as a template to insert a G, or you can have a C there, or you can have a T there, right? So 3 out of those 4 combinations would be wrong. There would just be that one that will, insert a C against that opening and restore the original wild type sequence. So just having that apurenic site within just 2 to 3 generations will give you a heterogeneous mix of bacteria where 3 out of 4 bacteria are, gonna have a random base inserted in that place. Okay, so 75% within a very brief period of time can begin to display a different genotype, because you have, if this was within a gene, then you change the structure of that gene. And if this was a critical gene, then you might also have the associated phenotype with it. Okay, so you can have spontaneous mutations that occur within cells all the time. Fortunately, cells have proofreading mechanisms that prevent them from accumulating these mutations. And mutations arise, but they're corrected. And so they aren't always passed on unless cells have some sort of a mutation within their proofreading genes. Okay, questions on that before I move on to induced mutations? Okay, all right, so induced mutations are mutations that we will basically carry out in a laboratory setting. We're trying to maybe have this random mutagenesis event where we're trying to induce a single mutation in cells. So we're not going to be able to control what mutation that is, right? So by random, it means that some random gene would be mutated, but we want just one gene to be mutated per cell. You can't have multiple mutations occur within individual cells, because then that makes it harder to attribute the resulting phenotype to those mutations. You don't know, which one, is responsible for the phenotype you see, or is it the combined effect of all of those mutations that is resulting in the phenotype you're seeing? So in scientists in laboratory will use different types of mutagens, and you have, a bunch of mutagens that are listed here. They can either be base analogs, so something similar to what you're seeing with the enol and amino forms of bases. These are just going to miss base pair, and miss base pairing, if retained, can of course result in stably passing on those changes. And so, it can result in mutations. You can also have a similar phenomenon where, there are agents that will add methyl groups or alkyl groups to existing bases, changing their base pairing pattern. And then that results in misbase pairing. And then, if that's inherited, would result in stable mutations as well. So there's a bunch of these different mutagens that can be used. When I was working in a genetics lab, we used UV light. So we would expose our yeast cells to UV light for a very brief period of time, because the longer you expose them to UV light, the longer the cells had to mutate, and you can have individual cells with multiple mutations. So you have to really control the amount of chemical mutagens or physical mutagens in a way that it would just result in a single mutation per cell. Okay, so there are different types of agents. All of them are going to essentially result in disruption of the base pairing abilities of the template DNA. So here you're seeing the N-methyl N-nitrosoguanidine, which adds a methyl group to guanine, and it changes its base pairing abilities because it used to use this carbonyl group to base pair with cytosine so that it could form 3 hydrogen bonds. In this case, with that oxygen gone, and now it's methylated, it no longer can take part in that hydrogen bonding with cytosine. So now it's no longer going to hydrogen bond with cytosine, and instead it's going to base pair with thiamine. And of course, then that can, on a subsequent replication, result in mutations. So you can have these other chemical compounds that act similarly as well. And to give you an example of physical agent and how UV light works is that UV lights generally would take pyrimidines. It doesn't always have to be thiamine dimers. You guys have talked about this in lab. We used UV light to control the growth of bacteria. The longer the exposure, right? The greater the rate of cell death. And that's because a lot of the pyrimidines, especially thiamines, if they tend to be in neighboring areas of DNA, they covalently link with one another. And once they do that, then they are no longer really available during the replication process for the copying of the DNA. And if replication of the DNA cannot occur, then you cannot, of course, have 2 copies to pass on to the daughter cells. And so there's really no need to undergo, cell division. So cell division stops and eventually the cell dies. And that's how UV light controls microbial growth. So, UV agents can be used as well, but you have to be very careful with the exposure time, and just so that you have this occur at a very, very small rate within cells. Now, in terms of, we're going to come back and we're going to talk about how cells may have the potential to go back and correct these mutations. If we're trying to introduce these mutations intentionally into cells to study the function of genes, then we want to ensure that our cells are lacking gene repair and proofreading mechanisms. Okay, it's not necessarily the DNA polymerase proofreading mechanism that should, of course, be there, because in absence of that, too many new mutations would be present. But we can certainly have some of the proofreading mechanisms absent, so that once, say, a mutation like with the nitrosoguanidine has been introduced, cells don't go back and correct it before the next replication cycle. Now, what happens when mutations occur? So as I mentioned earlier, if they happen within the coding region of a gene, they're going to result in change of the codon. And depending upon the change to the codon, you can have different results. So if we look at the original sequence here, it codes for 3, 4, 5, 6 amino acids. So there should be 18 bases there, and you can divide them into individual codons, starting with the ATG, because that's where your reading frame begins. This is your initiation codon. This is where we begin to read the codon sequence, and this is where the small ribosome sort of associates and initiates the addition of amino acids to the polypeptide. So we start reading from there, and then we can essentially continue to read the sequence in triplets, and we can consult the genetic code to figure out what the corresponding amino acids would be. Now, if there are changes that are made, okay, a mutation arises and it isn't corrected, it can result in newer codons. And sometimes these codons can be forgiving, and that has to do with the redundant nature of the genetic code, that you can have several codons that could code for essentially the same amino acid. So in this case, you have the ACC and ACA that are both coding for threonine. So this is sort of like a forgiving mutation. And so we refer to this as a silent mutation. It didn't really affect the protein structure because it did not affect the protein sequence. Then you can have what we refer to as missense mutation, so that the third codon that was TCC has now been changed into TGC. And so the original amino acid coded by the original codon was serine, and now it's been converted into cysteine. And so these are two completely different amino acids, and so they have the potential of interfering with the secondary and tertiary structure of the protein, and hence the function of the protein. So we refer to this as the missense mutation. Okay, then you can have another type of substitution where one base is changed for another, so that we have converted the lysine codon that was a triple A into a TAA. So this first A was substituted by a T, and the result is that this corresponds in an mRNA to UAA, and UAA happens to be a stop codon. So this is referred to a nonsense mutation, where this base substitution, this point mutation, resulted in conversion of a sense codon, meaning it coded for an amino acid, to a nonsense codon, meaning it does not code for an amino acid, but rather serves as a stop codon. So this would result in a shorter than normal protein, in what we refer to as a truncated protein, so that you could have just a part of the protein that's prior to the stop codon there, but anything that is after that is lacking. Okay, so these, of course, proteins would be non-functional because they are not, fully synthesized. Then you can have what we refer to as frame shift mutations. This happens due to the insertion or deletion of individual bases. So we can start comparing this sequence to the original sequence that is given, and we notice that the first codon is the same, second is the same, third is the same, right? And then fourth is the same, and well, if you have a G inserted before the fifth. Okay, so the problem with this is that the ribosome only reads these bases in triplets. It's going to start reading normally from the beginning, and then it arrives to this G, and it automatically makes this G pair up with these A's, so that the codon is read as GAA, and the next codon would be read as AGG. So these sequences are going to be, amino acid sequences are going to be no longer the ones that you were seeing initially. They have completely changed because the codons have changed, and this can have a dramatic effect on the length of the protein, because you can have earlier stop codons arise due to this insertional mutation, or you can have a lengthier protein because the stop codon has been removed to much later on. In either way, it's going to result in a dysfunctional protein, because the protein amino acid sequence or the primary sequence isn't what it was supposed to be. So you can have mutations arise from not just substitution, but insertion and deletion as well. Sometimes microbe will have a secondary mutation, sometimes at the same site, that helps them restore that original sequence. So you started off with the threonine codon ACC, which was converted into a triple C, and then it was converted back into ACC. So we refer to the second mutation as the reverse mutation because it helped to restore the original genotype and phenotype. You can also have mutations that this is just, I guess, another example of a forward mutation where TCC has been converted into TGC, and that changed the serine codon into cysteine. And then you can have the reverse mutation that might change, might not restore that original codon that was TCC. We have AGC, but fortunately, because of the redundancy of the codon, it restored the original amino acid that was encoded by TCC. So this is going to also restore the original phenotype. And then you can have mutations where a reverse mutation where the amino acid that is replaced is chemically similar in properties to the amino acid that it replaced, essentially. And so here we have a threonine, which is a polar amino acid. The forward mutation changed the ACC code into CCC. Proline is a hydrophobic amino acid. The problem with hydrophilic and hydrophobic is that hydrophilic amino acids tend to be surrounded by water, so they're going to be somewhere on the surface, and hydrophobic want to be away from the surface, so they tend to be towards the core of the protein. And so this could really bring about a drastic change to the structure of the protein because proline wants to hide itself. And so it can prevent the proper folding of the protein. Now, in this case, a reverse mutation has changed that code from triple C into CTC. In this case, you haven't restored the threonine, but you have replaced proline, which was a non-polar hydrophobic amino acid, into another polar amino acid so that it could, again, be found at the surface, and hopefully it will help to drive the same tertiary structure that we started off with, so they could be, forgiving. Sometimes you can have suppressor mutations. Suppressor mutations are mutations that will occur in a completely different gene. Reverse mutations occurring within the same gene, right, at the same site, whereas suppressor mutations are where you have a second mutation elsewhere. So say we started off with this frame shift mutation, and perhaps later on you have a second mutation that resulted in a deletion elsewhere. Okay, so originally, I'm so sorry, I don't like that this pointer goes away. But so you have the forward mutation where G was inserted and anything beyond that G was incorrectly encoded. But, say there was a suppressor mutation here. I guess this this is a suppressor mutation within the same gene, just at a separate site, so that it's earlier than that original insertion. You have a deletion sequence. I'm sorry, it just kind of goes away. We would have to see that this T is gone. Okay, so the third codon has now been changed from serine to proline, which again is a problem because you've gone from a polar to a non-polar amino acid. But at least the second deletion mutation has restored the amino acid sequence beyond this deletion. All right, so just the region between the insertion and deletion would be incorrect in terms of amino acid sequence, but everything before and after would be correct. Okay, and so originally, as I said, suppressor mutation that happens to be in another gene. You could have maybe a TAA recognized by a tRNA that encodes for lysine, right? So generally, there is no tRNA that would correspond to this codon, but there could be a suppressor mutation in the tRNA gene, which changes its anticodon so that it can correspond to TAA. and it happens to encode for lysine. So of course, those types of mutations are very, very rare, extremely rare, but have been witnessed within cells. Now, in terms of mutants, when you have mutations, then you have mutants. And so you can have different types of mutants. We can have morphological mutants. These are mutants that look different. So there's something about them that you could see phenotypically. It could be maybe color of cells, like you could have, Serratia marcescens cells that are either forming just white colonies versus pink colonies, right? So those could be morphological mutants, or you can have cells that have flagella versus no flagella. Right, so you can see that. Then there could be conditional mutants where the phenotype isn't immediately obvious because the mutations can actually be lethal. All right, so you introduce a change into a very critical gene, like say, several, a strain that only has DNA polymerase 3. If you make a mutation in that DNA polymerase 3, cell is not going to survive because that's all that it has to replicate its DNA, right? So how do you study then mutations of a critical gene like a DNA polymerase 3? Perhaps we could introduce changes in parts of the gene that are not critical to the function of that particular resulting protein at a given temperature. All right, so generally proteins or bacterial cells, I would say, they're going to respond to changes in, say, temperature. So you grow them at one temperature and you elevate that temperature, say you take them from 23 degrees Celsius to 37 degrees Celsius, they're going to initially have this denaturation of the protein that they're ready to correct because they have this heat shock response proteins that are beginning to express the chaperone proteins that will help to correct that initial denaturation. So you can have a mutation where that initial denaturation cannot be corrected by the heat shock proteins, for example. All right. So conditional mutations are generally, trying to express lethal mutations under certain restrictive conditions. Okay. And so generally you can have strains that are, for example, prototroph, going back to the example of DNA polymerase 3, this would be the strain that has a fully intact DNA polymerase 3. All right, so that it doesn't need any particular growth conditions, any particular nutrient in order to, fully grow. Then you can have in auxotroph mutation where that strain maybe is lacking the TRP operon or a critical gene within the TRP operon, so that it's unable to synthesize its own tryptophan. So you must supplement its growth medium with tryptophan or it's going to die. Right, so we call such mutants as auxotrophic mutants because they must get some sort of a nutrient in order to continue to grow, or they will, die. Now, going back to that same example of microbe with the DNA polymerase 3 mutation, if that mutation is in a critical part of the polymerase, so that the cell is unable to synthesize DNA at any temperature, then the cell would die. However, if the mutation happens to be in a not so critical part of the DNA polymerase required for its replication abilities, but maybe folding abilities, then you may be able to grow it at a restrictive temperature of, say, 23 degrees Celsius, and would call that the permissive, temperature, not restrictive. That should be the permissive temperature. And the restrictive temperature is where its growth would be restricted, which would be, say, 37 degrees Celsius. Okay, so I apologize, I misspoke. So there are so many different types of mutations that can result in the death of a cell, say mutations in critical enzymes of glycolysis, for example, can result in death of the cell because they need those enzymes to continue to able to synthesize ATP. So how do you study them? We generate conditional mutants, okay? You can also have, of course, mutations in regulatory sequences. Mutations don't always happen to be in coding regions. You can have mutations, for example, in the operating region of the LAC operon. If the operating region is mutated so that the LAC repressor cannot bind, then that LAC repressor would always be expressed, right? Transcription would always occur. But what if the mutation is such that LAC repressor binds but cannot come off? Okay, so then in that case, you can have a LAC operon which is completely intact, but it's never transcribed because the LAC repressor never goes off. Similarly, you can have mutations in the activator sites as well, or just the promoter site where the DNA, where the RNA polymerase itself binds, right? If it's unable to bind, there is mutations in the TATA box or mutations in the -35 sequence, then the polymerase can't bind. If polymerase can't bind, transcription cannot occur. Nothing wrong with the RNA polymerase, but its ability to bind is affected by the actual sequence that has been mutated. Okay, so you can have mutations in tRNAs, as I just mentioned on the previous slide. You can have mutations in your ribosomal, RNA genes as well that will help to, sometimes generate newer strains of bacteria. And when we're looking at we're comparing the 16S ribosomal RNA to compare lineages of different strains, that is where the ribosomal RNA sequences come in, and the how they have diverged over time. But of course, something like a ribosomal RNA and tRNA, because they are so crucial to the process of protein synthesis, you're not going to see a huge amount of divergence in the critical regions of the ribosomal RNA. So you're always going to have conserved sequences that are critical to the protein synthesis function, but there might be other regions that are free to diverge because they're not so critical to the function of the protein. So some mutants would be lethal and they would automatically die out. And there would be other mutants that would be stably, able to go on because the mutations are not in the mutations are not lethal to them, although they are still in the same gene. Okay, it's just how critical that region was to the function of this RNA is what you need to consider. So if we can generate mutations in the laboratory, then we want to be able to isolate mutants as well, right? So as I mentioned earlier, when I was studying, we would generate mutants that were mol minus mutants, and we wanted to ensure that there was only a single mutation per cell. And we exposed millions of cells to radiation for a very brief period of time. And not every single one of those cells was going to become mutant. And so there were going to be a few cells that were going to become mutant because of such low dosage. And we had to have a way of screening for that mutant, finding it out amongst a bunch of other cells that were still that original wild type that they hadn't become mutants. So how do you go about doing that? So we can increase the rate of mutations in a given cell by using some of these chemical or physical methods to 1,000 to 1 mutation per 1,000 to 1,000,000 cells. Okay, so that's you want to compare it to 1 mutation in 10 million to 10 billion cells. So this is quite a lot. And so we're going to choose an appropriate mutagen here. We're using nitrosoguanidine. And so we take our cells and we basically grow them in a growth medium that has been supplemented with a very minute amount of nitrosoguanidine. And of course, you want to grow these bacteria with complete medium so they're growing completely fine. And after that brief exposure, you take a little bit, you plate them onto a master plate, and we have the complete medium, so all the cells are growing here. So we do have a single mutant here represented here as red, but of course you can't always associate phenotypes with visual phenotypes with mutant genotypes. We would be very lucky to have something like this where you can visually just detect it, right? But in this case, let's assume that there is no way of visually detecting that mutant. So how do we then, take that particular colony out away from everybody else? How do we distinguish it? So one method is referred to as screening method, where we're going to basically screen all the bacteria that we initially treated with this mutagen. And this method is known as the replica plating method, where you have this large plastic block and you place a sterile velvet fabric on one end of it. You press it against that original plate, and that generates a impression of all of those colonies. Some of those colonies and some of those bacteria will transfer onto the velvet. And then you can use that velvet replica to make an impression onto several plates. So if you wanted to test 5 different conditions or just 2 different conditions, that's up to you. But you can go and just continue to stamp individual plates, all the plates that you need to, test different conditions for. So in this case, we have a replica plate that has the complete medium and a replica plate that's lacking lysine. So we're obviously looking for a mutant that can grow in absence of lysine. Okay, so these were all cells that were growing in presence of complete medium. They did not need to worry about synthesizing any of their amino acids. But here we're doing one stamping or replica plating on complete medium, so you should get exactly the same replica as the original master plate. And we're doing one in absence of lysine. We could have done one in absence of tryptophan, one in absence of alanine, one in absence of, whatever, maltose or whatever you're testing. And so in this case, what we see is that when we compare our replica plate for complete medium, it gives you basically the same pattern as you see in the master plate. But when you look at the replica plate that's lacking lysine, one of the colonies is missing. So you really have to sit down and visually inspect these plates until you can figure out the difference between the two of them. And the colony that is missing in absence of lysine is the colony you need. You will go back to the plate that had the complete medium, recover that colony, and now that colony hopefully contains a single mutation, and you can do further studying with that and figure out what gene it was that was missing that prevented this cell from synthesizing its own lysine. So that gives you a clue about where to look for genes that are involved in lysine synthesis. And so it might be a operon. So you find out all the genes that are involved in the biosynthesis of lysine. Okay, but of course you have to start off by planning your experiment carefully. You need to, of course, know what are the conditions under which your cells that you're interested in would grow. And you are going from a strain that was auxotrophic, meaning it didn't require any special nutrients, to a prototrophic strain. I'm sorry, you started with a prototrophic strain that did not require any nutrients to an auxotrophic strain, which now requires lysine in order to grow. And you maintain it. You can freeze them at minus 80 degree endlessly, and you can go back to them and continue to study them as you work on them. We can also do a selection, so where you don't want to look at all the transformed versus wild type versus mutant cells. You want to just look at the mutant cells, right? So there are ways of doing that as well. Again, it all has to do with how carefully you select your growth conditions. So the example that we're looking at is what we refer to as the Ames test. This test is done to test the mutagenicity of compounds. These compounds might be used, for example, as preservatives in food, or they might be used as stabilizers, say in makeup or in drugs. And so, generally we know that mutagens can cause cancer. So anything that is suspected of being a mutagen has to be further tested on live organisms like mice to detect whether they have the capability of causing cancer if we consume it. But this is where we, start off. The Ames test, basically we're taking a strain of Salmonella bacteria and we're growing them in presence of this chemical that you want to test. And so the strain of bacteria that we have are histidine auxotropes, which means that they require histidine in order to grow. So we're going to incubate our Salmonella in presence of a compound that we want to test. So we have the minimal medium, right, which has a small amount of histidine. It has everything else, but a very small amount of histidine so that the cells will run out of it pretty quickly. And then you have the same medium, but now also with the test mutagen. And what we see is that this strain has this habit of, reverting back to the prototrophic status when it comes to histidine, meaning it goes from being unable to grow in absence of histidine to some reverting mutants that can restore that ability to grow in absence of histidine. Okay, so this was sort of like a reverse mutation that's going on. So this is a strain that spontaneously does it. It kind of, flops back and forth between the auxotrophic and prototrophic state. And so what happens in the presence of the test mutagen is that you can have many more revertants. Okay, so this is telling us that a strain such as this histidine auxotrope is able to restore its prototropic status at a much higher rate in presence of this chemical compound. And so therefore this could be a suspected mutagen, because in presence of the mutagen, the rate of mutations is higher. In this case, they are reverting back to being wild type, but it still is a mutation, right? If this mutagen had no effect, then you would basically see the same number of spontaneous revertants here in presence of the mutagen as you would in the absence of the mutagen, but you're not seeing that. You're seeing a much elevated rate of revertants, so you can it's sort of like a signal for you to study this mutagen further. Now, in order for Ames test to work, we're gonna use this strain because one, it was the auxotropic form, so it needs histidine, and then it forms these reversion mutations, but it also has cell walls and membranes that are more permeable. So all of these chemicals that you're testing, they're able to enter the cell and introduce these mutations. And the mutations, once they're introduced, should not be repaired. So these are strains that are going to lack these DNA repair mechanisms. And, generally these chemical mutagens, especially if we're suspecting them of being carcinogenic, you have to treat them under conditions that would be similar to what they would encounter within the human body. Often any nutrients that we take in generally would go to the liver first, where detoxicity would occur or some manipulation of that chemical compound would occur. And that can result in either this mutagen remaining intact and being released as intact mutagen to be picked up by other cells to cause cancer elsewhere, or it can be just detoxified and it's just released as something that is no longer mutagenic. So oftentimes in this Ames test and to this growth medium, you would also have some sort of a liver extract that is added to replicate those conditions of what happens within the human body. Okay. So, you could have the liver extract, for example, just break down the mutagen, and the result is that you would have the rate of reversion similar to what you would see in spontaneous reversions, okay? But in this case, notice the growth conditions were such so that there was very little histidine. So initially cells grew using that histidine, and once they realized that the histidine was gone, anybody who didn't have the ability to synthesize their own histidine died out, and anybody that possessed the ability to synthesize their own histidine survived. And so, this is a selection for the prototropic forms of this particular strain, okay? Questions on any of this before we move on? Okay, all right, so let's move on. We're gonna now talk about some of the DNA repair mechanisms, as I mentioned initially, that these mutations are extremely rare. That doesn't mean that the actual changes to the genome never happen. They happen, but bacteria possess DNA repair mechanisms so that they go back and correct those mistakes. And one of the first ones is the DNA polymerase itself. The DNA polymerase 3 has the epsilon subunit that it uses for proofreading from 3' to 5' for sliding backwards and testing and removing incorrect base and replacing it with the correct base before moving forward. But sometimes that doesn't happen. Sometimes errors will be made. And so how does the cell then go back, scan the newly synthesized daughter strand, and ensure that the correct bases have been inserted opposite the original parental strand that served as the template strand? So we have what we refer to as the mismatch repair, where mismatched bases would be basically detected and replaced. So you have these 3 proteins, MUT-H, MUT-L, and MUT-S, whose job is to kind of bind the DNA, glide along it, just slide along it, and continue to look for any mismatch pairs. So MUT-S's job is to continue to recognize any mismatch base pairs. So T and G here are mismatched, right? But how does the cell know which is the correct base? Was it G that was correct? Was it T that was correct? So cell has to somehow know what was the original parental template strand. And if it knows what was the original template strand, then it knows that the base opposite that is incorrect. And so it replaces that base instead of the base that you would find in the parental template, right? I mean, it makes no sense for a cell to come back and remove the base from the template and against it place an A in this case, and that would be a stably inherited mutation, right? We want to remove the T and replace this with the C, because here we know that this is a parental strand and this inner strand is the newly synthesized strand. But how does the cell know that, right? How does the cell keep track of it? So cell has this system of methylating its DNA. So E. coli, for example, have DNA methyltransferase, or DAM. What it does is that it's also interacts with DNA and it looks for these sequences, okay, GATC. And every time it comes across the GATC, it methylates the A, okay? So it happens on if this was original DNA that had, that's not undergoing replication right now, both strands would be originally methylated, right, on all of these GAT sequences. So when the DNA undergoes replication, the newly synthesized strand would not be methylated for some time. So this DAM enzyme is going to be slower than the replication machinery. So it's going to take some time before it catches on and it methylates all the GATCs. But this small window between replication and methylation is what the system needs. This system of mismatched repair needs to differentiate between the methylated parental strand and non-methylated daughter strand. So we refer to this state as the hemimethylated state. Half of the DNA, in this case, one strand is methylated, the other isn't. So MUT-H's job is to recognize which one is the parental strand based off of that methylated state. Once it recognizes that, then it's an endonuclease, which means it can cut the phosphodiester backbone. And it's also a exon, it's not actually itself going to do it, but it's going to hire an exonuclease to come in and just chop up. Okay, kind of like Pac-Man. I like to think of like exonucleases as Pac-Man. They come in and they eat away at all the bases. So the chopping is going to start from one methylated site where that GATC is, all the way to where that incorrect base was detected by the mutase. And then you're going to have the DNA polymerase come in and fill that in. So generally it's DNA polymerase 1. So this function is very similar to filling in gaps where the primers would be. Okay, so you're going to have DNA polymerase 1 come in and then, fill this in with the proper bases. And then eventually the second strand will also be methylated. And of course, this mismatch has been repaired. Okay, so sometimes when we're trying to induce mutations, like we just talked about induced mutations, we use strains that have a mutation in the DAM gene, so they're DAM minus, okay? Because if they have this mismatch repair system, they will go back and correct the change that we just introduced. So mutational analysis requires that cells lack this particular, mechanism, and also sometimes introducing new pieces of DNA requires that this DAM mechanism be absent as well, because bacteria use this methylated state as a means to know what's theirs and what's not theirs. And the reason they want to know that is because they're undergoing infection by bacteriophages. And this helps them recognize their own DNA against a viral DNA. And if it's not methylated, their first response is to chop it and throw it away. Okay, so you can understand, hopefully, the implication and importance of all of these players in mutational analysis. Second mechanism of introducing repairs is what we refer to as the excision repair. And there are sort of two types here. Change is really, again, at a smaller level, they're named different things because you have different enzymes that are carrying out these reactions. So in nucleotide excision repair, you have a system of endonucleases called UVRA-BC. UVRA and B, they kind of like what we just saw with the mismatch-based repair. You had, mute S kind of scan along the DNA looking for mismatch bases. Here, you have the UVRA and B that scan along DNA looking for any type of really damage, but particularly dimers, so UV damage particularly. And this is why you see the name UV as part of these proteins, because they're involved in UV based repair. And so you will have this recognition of the thymine dimer, and then the interaction between the A and the B changes so that the A leaves and UVRC joins. UVRC happens to be an endonuclease, so it's going to make a cut in the backbone of this DNA strand on either side of that pyrimidine dimer. And once that cut has been made, then you can have UVRD that is not shown, that is a helicase, comes in, removes this intermediate piece that's no longer joined at the backbone to the rest of the DNA, and DNA polymerase is going to come in, fill in this gap, and DNA ligase is going to come in and seal that gap. All right, so that thymine dimer has now been replaced with just simply, thymines. Then you can have base excision repair. And in base excision repair, again, it can correct any type of damage as well. It could be, chemical changes as well, not just UVR-based changes or UV-based rather changes. And here you have different enzymes such as glycosylases whose job is to recognize, sites where you either have a wrong base that's inserted. So you're not supposed to find a uracil within DNA, so that needs to be removed. And then you have this apurenic site that needs to be, replaced. And, in order to do that, there is no enzyme that's going to come in and just add the nucleotide, change the structure of the nucleotide by adding the nitrogenous base. So you still need an endonuclease to come in and make a cut in the backbone, remove this small area that has this base missing. DNA polymerase 1 works together with the AP endonuclease and glycosylase again in this form of excision repair, as we saw in the nucleotide excision repair as well. It's going to come in and it has the ability to act as that little Pac-Man to kind of chew away and get rid of not just this site, but a few bases adjacent to that. And then it's going to use its 5' to 3' polymerase activity to fill that in. And ligase comes in and just seals the backbone so that you have one intact DNA strand. So you have at least 3 systems that E. coli has, where it comes in and it tries to repair damage to single nucleotides. But what if damage is at a larger scale, so that there is a large piece that is missing, or you have a large piece that is missing against another mutant site? How does a cell correct that? So fortunately for bacteria, especially rapidly dividing bacteria, we know that they're always going to have, although they are monoploid, they're going to have DNA that is undergoing replication already. All right. So there are some bacteria that are polyploid, and so they can have multiple copies of the genome, and so they can do recombinational repair by using, the intact copy, recombine it with the damaged copy, and then repair both. But in this case, where you have just a single copy of the genome, if this cell is already undergoing replication, then it's going to have, fortunately, another strand where it might be intact, so that it is able to swap a region of the parental strand from a sister DNA strand, and just swap one region, correct that first, fill that in with DNA polymerase, no problem with that. You have an intact template against that, no issues. And then, have the UVR-ABC system come back and remove these thymines. So you can have this recombinational repair that happens with the help of a particular protein called REC-A. If you don't have the REC-A protein, then the recombination doesn't occur. So this is how important it is to the repair that involves swapping pieces of DNA from one DNA to a different DNA strand. Okay, but so you have this sort of repair that would occur on both of these strands eventually, and the bottom strand is still dependent on other proofreading mechanisms, namely UVR-ABC system, to correct this thymine dimer. Okay, but they can correct that as well. What happens if the cell is very, very damaged, so damaged that it can't rely on one of these systems? Then the cells begin to express all of these systems all at once: mismatch repair, the excision repair mechanisms, and the recombinational repair. REC-A would be present, UVR-ABCD system would be present, the AP enzymes, and everything else that we've talked about, all of them will be expressed. And often within bacteria, as we talked about in regulation chapter last week, you're going to have regulation of transcription. So there was going to be some genes that are going to be active all the time to kind of oversee the normal replication cycle. And there are others that are going to be expressed under extreme conditions, and their expression is controlled by controlling transcription. So if you could control transcription by employing a repressor, then you can control expression of a bunch of different proteins all involved in this SOS response, as we call it. And so bringing in the repressor shuts off all of these proteins together. Removing this repressor is going to turn on all of these proteins together. So, kind of like what we talked about with different sigma factors, right? So here it's not the sigma factors, but rather individual repressor protein. And that repressor protein is referred to as the LEX-A protein. LEX-A protein is a repressor protein, binds to a bunch of different promoters and turns off transcriptions of all the corresponding genes. And under SOS response, we have the REC-A protein that actually destroys the LEX-A repressor, and that basically turns on expression of all of these, genes that can help to basically allow cells to survive. The goal is not a repair of like individual bases and single nucleotide mutations. Their goal right now is retention of as much of the genetic code as possible. And then, survival. Their goal isn't replication. Their goal isn't passing on this genetic material intact to the daughter cells. Their goal is, if I can retain enough of this information so that I can continue to synthesize enzymes to survive, then that's all that they're looking for. So in fact, as part of this response, you're going to have this protein called SFIA that is going to prevent cell division, which is important, right? You don't want to be passing on all of these mutations to the daughter cell. So these types of mutants grow very, very slowly, if they ever grow at all, because it will mean their growth and their cell division means passing on a bunch of mutations to the subsequent generation, which might not be able to survive. And so you can also have production of newer DNA polymerases who are more so involved in recombinational repair, especially if DNA has been, the backbone has been fractured in many different places and you have to kind of put the DNA pieces together. You have E. coli specifically has additional DNA polymerases, DNA polymerase 5, DNA polymerase 4, that have the ability to kind of ignore the damaged areas and kind of randomly just insert something against those damaged areas, and then continue to replicate the intact DNA and trying to salvage whatever they can, okay? But of course, as I speak about it, of course, I hope you understand that this will result in numerous mutations that would be introduced to bacterial genome as a result. Question. Cell doesn't want to have an SOS response unless it's needed, unless it's in a life or death situation, okay? So if cell has had so many mutations, a lot of damage to the DNA, then how does it ensure its survival? So that is what the SOS response is really. Okay, so it doesn't want it. This is why it has the LEX-A repressor that's kind of preventing all of these proteins from being expressed unless absolutely necessary. Anything else? Okay, so another important phenomenon in bacteria is the process of horizontal gene transfer. So generally, we're all aware of the vertical gene transfer, where genes will be transferred from a parent to the daughter cell. And of course, that's happening within bacteria as well. But we know that bacteria are also able to share the genetic material, right, from a person to person, and so, or from a cell to cell rather. And that's just not something that we see in multicellular organisms, right? Multicellular organisms can contribute their haploid, genome towards a zygote, and then that zygote will produce a whole new organism. But, you don't pass on your genetic material to each other. And so bacteria can have this diversity and the prokaryotic diversity that we see. You can attribute it mostly to horizontal gene transfer. And there are multiple ways this can happen. Often viruses are the major reason behind this gene transfer between prokaryotes, and viruses are the major evolving force in prekaryotes as such. As we said, there's going to be plenty of spontaneous mutations, but not enough, right? And these spontaneous mutations are going to be corrected as long as the cell is not really under very stressful conditions. It's expressing its normal repairing mechanisms, and it will repair those, right? So then how in a natural setting you can have such enormous variety, and it turns out that viruses are the largest vehicle for evolution in prokaryotes, in cells in general. And so what we're saying in this case is that you can have a bacterial cell that picks up this DNA, this exogenous DNA from some source, and it's going to pick it up, bring it inside, and it can either integrate that newly internalized exogenous DNA into its own chromosome. So every time chromosome is copied, so is this DNA. And so it will be passed on. So it will be stably inherited, and you are going to have recombinant cells result as a result of this single bacterium that has picked up this piece of DNA. You can also have a piece of DNA that has its own origin of replication, and it can form its own little circular piece as a plasmid. So it's copied, even if it's not copied as part of the bacterial genome, it will be copied because it has its own origin of replication. So every time process of replication occurs, the exogenous piece would be copied along with the endogenous piece, and you would have, it being passed on to both of the daughter cells. So again, it would be stably inherited. However, if the reverse is true, if the donor DNA cannot be inserted into the genome or cannot be autonomously replicated, then it will not be passed on to all the daughter cells. It may be passed on to one daughter cell, right? But it will not be passed on to the rest of the daughter cells. So it will be lost eventually. And as I was mentioning earlier, that every bacterial cell has a way of recognizing its own DNA against some exogenous piece of DNA. And unless it recognizes something about it, such as a methylated A in a GATC sequence, it's going to degrade it. All right. So if this DNA is too different, it's from a strain that is, too different from the strain that has picked it up, then it will just be degraded. And of course, it will neither be replicated, none of the genes would be expressed, and it would eventually be, of course, lost, even in the original cell that picked it up. Okay, so cells, when we talk about horizontal gene transfer, we're talking about bacterial cells that are able to have a stable recombinant result as a result of picking up that piece of DNA. So there are several ways large pieces of DNA can move about within the genome. One is transposable elements. These were originally actually discovered within eukaryotic organisms within corn, but then eventually we found its equivalent in basically all types of cells. And so transposable elements are basically pieces of DNA that will begin to move about within the genome. So they can be present in site A at one time, and then they can be present at site B at another time, or they can just excise themselves and are completely lost from the genome altogether because they never made it to site B. So they sort of change their appearance within the genome of cells. And so there are different kinds. Generally, they require some sort of an enzyme that helps them insert themselves into the genome. So not only go into the genome, but also pull themselves out or copy this piece of DNA sequence and then have that insert somewhere else, as we're going to talk about it in a minute. But the simplest of the transposable elements, or these jumping genes, as we call them, are what we refer to as insertion sequences. So insertion sequences have a transposase gene, which helps it code for a protein that helps to insert this insertion sequence into DNA. All right, and this insertion sequence has on either end what we refer to as inverted repeats. And we talked about what inverted repeats are. There are, sequences that can be present on both DNA strands, right? And they read the same from 5' to 3' direction. So you can have inverted repeats on either side of those transposase genes. And then outside of that are direct repeats. These are sequences that the insertion sequences, so this is DNA sequence that this moving gene recognized before it went in. Okay, so it just doesn't randomly go in. It recognizes a particular sequence that we refer to as direct repeats, and then it inserts itself within those direct repeats. So you can have insertion sequences, and then you have composite transposons. So composite transposons are basically two insertion sequences, insertion sequence 1, insertion sequence 2, both with their transposase gene, and the two are flanking a antibiotic resistant gene. So this is a composite transposon. So antibiotic resistant gene is moving as part of this sort of structure that we're referring to as composite transposon. Antibiotic resistant gene doesn't have the ability to move about unless it's flanked by these ISs that have their transposase genes, and they're still going to recognize the direct repeats and go into just that region where that sequence is present. Then you can have unit transposons as well. They can also have, genes other than transposases and resolvases and integrases and things like that, but they are not flanked by complete insertion sequences. So insertion sequence, remember, is 2 IRs and a transposase gene. And so you don't see that. You see 1 IR and 1 transposase gene, but you don't see the second IR sequence. Okay, so they're slightly different. Now, how do they introduce mutations? So there are two ways that these transposable elements move about. One is the simple transposition, where you have a transposable element simply excise itself, cut itself out of the original sequence that it was in, and pick itself up and then move to another site on the same genome. And when it moves into the new site, it could move into middle of a coding region and it could introduce a mutation there. Okay, there is another method which we refer to as the replicative method. In replicative transposition, you're going to have replicases. So there's going to be some gene that is encoded by the transposable element itself that's going to help copy this transposable element and then target it for its insertion into another site on the same genome. And again, you understand that this will result in a mutation if it was in a promoter, if it was in a terminator, if it was in a operator region, if it was in a coding region, right? If it was in the region that codes for tRNA, right? So anywhere it goes in, it will introduce a mutation. It's a large insertion. Now, even when sometimes this simple transposition occurs and the transposable element cleaves itself out and goes into a brand new place, it leaves behind traces of having transposed there, having lived there. And that can of course result in incomplete restoration, I would say, maybe not restoration at all of the function of that original gene. So just to help you understand that, we can have a transposable element represented by this red block, flanked by the inverted repeats. And as I said earlier, when the transposable element goes into a sequence, the target DNA has to have that sequence that we refer to as direct repeats on the prior slide. And so notice it's again, ATGCT on one strand, 5' to 3', and AGCAT on the second strand, going in the opposite direction, so quite similar. And when the transposable element goes in, it basically makes a staggered cut. The transposase makes a staggered cut, generating sticky ends, and then it kind of plops itself in the middle of those staggered ends. And so this basically results in duplication of this sequence on either ends of the transposable element, and that is what makes the two direct repeats on either side. So original sequence, right? The original sequence here had a single direct repeat, which was this sequence. After the transposable element went in, there are two of those on either side. And so when this transposable element picks itself up and then moves out of this area, these direct repeats are not corrected, okay? So there will be that addition of 6 bases here at this site, which will basically be a history of this transposable element having inserted itself here, as well as it's a mutation, right? It's an insertional mutation. Fortunately, if you make insertions or deletions in groups of 3, right? So in this case, we have 2 groups of 3, it can be forgiving. So before these 6 bases and beyond the 6 bases, if this was a coding region, everything would be correctly coded and protein would be correct. But within this region, you would have addition of 2 new amino acids that don't belong. And sometimes they can be enough of a problem that they can prevent the function of the protein, okay? But some of these, in fact, majority of these transposable elements would leave behind a trace of having been there. Questions on transposable elements? Okay, all right, so let's talk about the process of transferring plasmids. We've studied plasmids in lab. This week, you guys are doing the transformation process in lab, and you're doing chemical-based transformation where we're forcing cells to pick up DNA in within 45 seconds because we introduce holes within the membrane. We make the membrane porous and it allows DNA to rush in, and then we close the membrane pores immediately so that the DNA is trapped within the cells. But you know, this passing on of the plasmid occurs in bacteria under normal conditions as well. And we know the process of conjugation where the competent sort of bacterial cells, those that are able to undergo conjugation, are physically linked together via this tunnel of pillin protein that helps to transfer DNA. And so this ability to conjugate is based off of this plasmid referred to as an F factor. F factor is about 100 kb in size, and it has an origin of replication. So it can replicate itself autonomously outside of the bacterial genome, but it also has the ability to insert itself into the bacterial genome. Okay, so it's referred to as what we call an episome. It can exist exogenously outside of the genomic DNA, or it can insert itself into the genomic DNA as well. The genes on it, especially in operon called tra-operon, short for transfer, it codes for, you know, a bunch of these genes that that help bacteria during this conjugation process. So F factor is short for fertility factor. This is what allows bacteria to horizontally transfer DNA to one another. Okay, generally, transfer of DNA is from the bacterium that has the F factor or F plus to a bacterium that lacks the F factor or is F minus. So when you look at some of these genes, you don't necessarily need to know all the genes and their corresponding products, but you should know that these genes are encoding the pillin protein that will help to make this little tunnel that will link the two together. It will help to make a secretory system at the surface of the F plus bacterium to allow DNA to be pumped into this tunnel. And so you have proteins that make up what we refer to as the type 4 secretory system. There is other proteins that help with the, again, transferring of the DNA, as we're going to look at on the next slide. So as I was mentioning earlier, this F factor can either exist as an individual plasmid, or it can insert itself by recombination into the bacterial genome. So here initially you're seeing the bacterial chromosome, here you're seeing the F factor. F factor has an insertional sequence that it uses to basically recombine with the bacterial chromosome, and it will become integrated into the bacterial chromosome. And this will basically have now bacterial chromosome would have essentially two origins of replication, one that came from the F factor and the one that was part of the original bacterial genome. Now, once this F factor is found within the cytosol, whether part of the chromosome or not, we're going to look at both situations. First, if it's outside of the bacterial chromosome, the genes on the F factor will be transcribed and translated, and the resulting proteins would be used to synthesize this sex pilus, made up of the pillin protein. And this pilus over time shrinks so that it brings the two cells close together, because it helps to make the distance that the DNA has to travel from the F plus cell to the F minus cell a lot shorter. Okay, so you have this original, linking together of the two cells using the proteins encoded by the F factor. Then you have as part of this structure, you have the type 4 secretory system that is going to help the DNA transfer from F plus cell to the F minus cell. And what's transferred is the F factor in this case. And in order to transfer it, the relaxosome, which is also encoded by, you'd remember, relaxosome was encoded by the TRA1 or relaxase, but it's in combination with multiple other TRA proteins. But so in any event, so it's going to make a cut in one of the two strands of the F factor, and it's going to begin to sort of unravel the DNA so that you have one of the strand that peels away from the intact circular strand. And so that's the job of the relaxosome. Cut it, okay, make a cut or a nick in the backbone of one of the DNA strands, and then begin to replace that strand. And the strand that has or one of the two ends that have the 3' end can then be used to use as a primer to add further nucleotides. And so you're going to have this growing point from the 3' end that's going to help replace. So it's going to continue to synthesize new DNA, and that's going to continue to peel away this outer DNA strand. And so we refer to this method of replication as the rolling circle method of replication. A lot of viruses employ this as well. It allows cells to synthesize multiple copies of the DNA in a very short period of time without having to employ like primases and all of that. But in any event, so in this case, the F factor is really large. It's 100 kilobase pair, so it's going to take a while to copy all of that. And you want about 1 unit length, okay, one entire piece of the outer DNA strand, then transfer to the the cell lacking the F factor of the F minus cell. And so that's the job of the coupling factor. Coupling factor helps to associate the relaxase with the secretory system, and the secretory system is going to then have, pumps that are going to use ATP to really transfer this DNA from the F plus cell into the F minus cell. So as long as the two cells remain attached together, the F factor will continue to be pushed in. And it takes about an hour for the entire F factor to be transferred to the F minus cell or the recipient cell. And once it's within the recipient cell, then the recipient cell will use its own DNA polymerase to copy it into a double-stranded F factor. So you went from one of the cells being F plus to both cells now being F plus, and both cells having this ability to conjugate. Of course, the circulatory system is really complex. I'm not asking you learn all of this, but I just want you to notice how all the different TRA proteins of the TRA operon would be involved in helping to synthesize this secretory system that goes across the two membranes in the periplasmic space and so on. So this is when the F factor is external to the genomic DNA. What happens when F factor is actually inserted into the genome? Once it's inserted into the genome, it can take part in what we refer to as the high frequency, recombination. And so we refer to this conjugation process as the high frequency conjugation. And so in this case, the same process that we mentioned in the prior slide is going to happen, but as the relaxosome comes in and makes a cut into the one of the strands of the genomic DNA, and it begins to unravel it, it's not going to simply just unravel the F factor corresponding DNA. It's going to begin to unravel the genomic DNA as well. So what's being transferred is essentially a portion of the F factor, but a lot of the genomic DNA from the bacterium as well. So the F factor generally goes into a region that corresponds to, in E. coli, into a region that corresponds to these two genes, all right? So the Pro plus and the LAC plus means that these two genes are wild type in this original donating strand or the donor strand. And so in this case, in the recipient cell, this recipient cell was mutant for both of those genes. If one of these genes were able to be transferred during this process, then you can have a recombination between this LAC gene and the LAC gene that you have on the genomic chromosome, resulting in this brand new recombinant bacterium that now is no longer, mutant for both genes. In fact, it has acquired a wild type version of the LAC operon, for example, from the donor cell. So the recipient cell never received the whole F factor. It never became fertile. It doesn't possess the ability to conjugate with subsequent, cells. It can undergo another conjugation reaction with an F plus cell, but it itself is not F plus and therefore cannot conjugate with, F minus cells. But as a result of this process, it has acquired a wild type gene and it has acquired a brand new phenotype. So if you sometimes take, bacteria that have the F factor inserted into their genome and you have them sit with, recipient cells, some of the recipient cells will display a brand new phenotype apart from becoming just the, fertile, or apart from just having the F factor. Now, depending upon how much of the genomic DNA is transferred, you can have cells that can transfer just one of the two genes, or you can have, cells that can acquire both of those wild type copies of the gene so that they probably remained attached for a lot longer. And the longer they remain attached, the longer the DNA would be transferred and the more of wild type genes they would acquire in this process. Okay. Questions on this? Okay, good. All right, so bacterial transformation, as I said earlier, you're doing it in lab because we're kind of forcing the cells to pick up bacteria by transiently producing holes within the membrane using heat. But bacteria are able to pick up just exogenous pieces of DNA from the environment, bring them in, either degrade it because they recognize it to be too foreign, or they recognize it to be similar to their own, and they recombine it with their own genome, as we just saw with the F factor, and then they will continue to retain it, continue to copy it and pass it on to the daughter cells. Or you can even have, exogenous pieces like DNA plasmids be picked up and then, those are maintained as well. Generally speaking, within bacteria, that we're not forcing to pick up. We're not chemically transforming in laboratory. They have a very low ability to pick up DNA, and there will be different times within their growth pattern where they will pick up this DNA. And that process of having that ability to pick up DNA is referred to as competency. So a cell that is able to pick up DNA is a competent cell. And so depending upon the microbe, they can be in different parts of the cell cycle. Generally, when we talked about the stationary phase, we said that, a Streptococcus pneumoniae is going to begin to, be really competent when there are a billion to 10 billion cells versus E. coli or, some of the other laboratory microbes, they're going to pick up DNA at the early log phase. So you have to transform them at much lower cell density. When we look at the bacterial DNA pickup in under normal setting, DNA is a large, complex macromolecule, and it's not just going to diffuse inside the cell. One, it's charged; two, it's too big, right? And so you need to have some sort of a transport mechanism to bring in DNA. So bacteria that pick up DNA, they have this DNA transport system, and you're seeing two separate transport systems. Here's the one from N. gonorrhoeae, and here's one from the B. subtilis. But just to focus on the top one, you're seeing that there are different proteins that help to form a pore in the outer membrane versus proteins that form a pore within the inner membrane. And then there is proteins, these yellow proteins that help to kind of form this channel along the periplasmic space so that the DNA is able to pass through the periplasmic space without being digested by the enzymes within that area. And there's actually an enzyme here labeled nuclease that is going to help degrade one of the strands of the DNA so that a single strand is going to enter the cell, kind of like what you saw with the F factor, right? That you had a single copy. You didn't have the double-stranded DNA, you had the single-stranded DNA that was donated to the recipient cell, and then the recipient cell converted into double-stranded DNA. So the same thing would happen here as well. You need to have this expression of this DNA transport mechanism in order for cells to pick up DNA from the external environment, perhaps from dying cells that have lysed and released their DNA, and then break it up so that it's a manageable piece. And then as they are using their DNA system to take it up, they're going to degrade one of the strands and just import a single-stranded piece of DNA, and they're going to see if it's something that's similar to their own DNA, or is it something that is similar to, viral DNA. If it's similar to viral, they're going to degrade it. If it's similar to something that they have a homologous region somewhere on the genome, they're going to perform this homologous recombination. And then they're going to have, essentially in this case, a different phenotype that went from inability to use lactose to now having the ability to use lactose. Any sort of mismatch there might be that could be repaired by, of course, mismatch repair systems. Okay, so transformation is a mechanism of picking up just pieces of DNA and just kind of retaining it. Last concept is, again, something that you were aware of. It's the concept where viruses are going to infect cells and be a vehicle to transfer DNA. We talked about it when we talked about the rise of antibiotic resistance. We said that viruses, especially that are virulent viruses, meaning they only undergo lytic cycle, they will infect a bacterium, and as a result of their infection, they generally chop up the bacterial DNA because they don't want bacterium to continue to mount a war against the virus replication cycle, or even try to recover from this assault, or try to continue to synthesize its own protein. So they want it solely for themselves. They're very selfish like that. So they come in and they break down the bacterial DNA into pieces, and as new virions are assembled, the only requirement for, or at least one of the requirements for filling of the capsids, the heads of these new phages, is the size of the DNA, right? Viruses, bacteriophages are going to be DNA viruses. And so they're looking for a particular size of DNA. If it's 45 kb that they pack, then any DNA that is that particular size can be potentially packaged. And sometimes those errors will be made if you have a 45, say, kb piece lying amongst these DNA strands, then you can have maybe one of the virions assemble and take with it the bacterial 45 kb DNA instead of its own genome. And when this virion goes on and infects the next cell, it's going to inject in it not the viral DNA, but rather the bacterial DNA. And if it's useful to the bacterium will use homologous recombination to insert it in its own genome and keep it that way. So you can have this now brand new recombinant bacterium result, which originally it was mutant for both his anabolic operon and lysine anabolic operon. And now after this transduction event, you have the phenotype of these cells change to that it's no longer auxotropic with respect to the histidine operon. It can grow in absence of histidine as well. All right, so we refer to this as the generalized transduction method. Then you can have specialized transduction. And specialized transduction is similar to what we saw with the high frequency transfer and the conjugation. So in this case, we're looking at lysogenic viruses. So no longer just virulent viruses. Virulent viruses only have a single life choice. They go in, they kill the cell, and they leave. But lysogenic viruses would insert themselves into bacterial genome, remain there until they're induced to leave. And when they're being induced to leave, sometimes they will leave where they not only pick up the viral DNA, but they actually pick up some of the bacterial DNA as well. And in the process, because viruses can only package a certain size of the DNA piece, some of the viral DNA is left behind, right? So it's a recombinant piece, basically half viral DNA and half bacterial DNA. And so that's copied, new viruses are assembled, all of these viruses are defective, they will go on and they will attack further hosts, but those hosts are not going to receive the entire genomic instructions of the virus, so the viral replication would not occur, but some of that bacterial DNA that has been injected will be recombined with the genomic DNA and would be stably inherited. Okay, so you can have this specialized transduction that results in often passing on of specific genes. Just like with the F factor, you had often the passing on of, say, the proline gene or the LAC gene. Here, when the lambda, which is a virus we'll talk about next week, and it's a E. coli virus, and it has, it's a lysogenic virus. It comes with these sites called ATT that it uses to recombine with corresponding sites within the bacterial genome. And when it inserts itself, it's sitting next to either the GAL genes or the biotin genes. And so if errors are made during the excision, then you can have either the GAL gene or the biotin gene become a part of this specialized transduction mechanism. So it's not going to be just any genes that are randomly passed on from the original bacterial cell to the subsequent host. It's often going to be either the GAL gene or the bio gene, depending upon how the mistake was made as the viral DNA was excised out. Okay, so we refer to this as the specialized transduction because you can have a specific portion of the bacterial genome be present in the subsequent host cells. I'm going to stop at this. Questions? I'm going to stop recording.
Recording stopped.
Questions, guys? Okay, so I just want to again point out that I posted the regulation lecture already on Blackboard, and I also completed the translation part of the lecture. I'm sorry, I was having a hard time recording, so it took me longer than it should have, but I have both of those recording in their appropriate folders, and I will also post today's recording in folder 11, I believe. And we have our final exam on December 21st. We will meet here together for one last time next week. We're going to talk about viruses. We'll talk about the Lambda bacteriophage and see how complex its lifestyle is. And we've so far been just kind of talking about it goes in, it destroys the cell, and that's it. But we're going to look at specifically a few examples of how does it decide to go between the lytic cycle and lysogenic cycle. So that's what we'll kind of focus on. And then we'll talk a little bit about coronaviruses and talk a little bit about the flu virus as well as the HIV virus. And so that's what we're going to do next week. So hopefully I'll see you guys here next week, but that's the last lecture. And beyond that, we'll have our lecture final on December 21st. So make sure to do all of your reading and quizzes before the deadline. Okay, I'm not going to be extending anything beyond the deadline. You have plenty of time to still do it. So make sure that you're taking your time to do it. So it's on the 21st, I believe. I'm going to make that announcement so you guys are all on the same page with respect to that. All right, thank you so much for those of you who showed up. Take care.